Good morning, everyone. And so my name is Deepak, and I'm here with LK, and we will be talking about bridging clinical and genomic knowledge in the context of Swiss Personalized Health Network. So when talking about health data, there are different types of health data, and they're captured at different levels and different settings. For example, you have data from a healthcare setting, laboratory, research setting, data that you have from your wearables and devices that you capture from home or patient-reported outcomes, and also administrative data like claims, billing, statistics, and so on. And each of these data is useful in one way or another, but collectively, they give you a bigger picture of what is the state of a patient and it gives an idea of their medical journey. And it is true that this data is useful, but also there's a need for data standardization and harmonization. And this is particularly true when you want to use this data for some applications where you want to get something more from that data and also understand a patient state and get them and also use it in personalized medicine, actually. And there can be various efforts that have been happened individually, like when researchers get access to data, they do it themselves, and it's kind of like a one-off harmonization. But this is not scalable, so there's a need to have a more systematic approach where you want to have interoperability of the data at the source. And this is particularly true in the case of the Swiss Personalized Health Network, which is a national initiative with the goal of making health-related data fair and also available for research within Switzerland in an ethically and legally compliant manner. And one key component of the SPHN is that we bring together various research institutions and hospitals together, and we encourage the secondary use of health data. And so when you look at the data that originates from the hospitals, especially clinical routine data, this data can be from various sources scattered across different departments like diagnosis, lab tests, billing, imaging. And there's a need to integrate all this data because we want to use this data and also we want to use this data for primary use and also promote its secondary use. And we also need to understand the meaning of the data, not just by the humans, but also the machines, because that's what we want to be doing, because is much more of a scalable approach. And we want to link this data to other sources. This could be data from omics, clinical trials, registries, sensor data, and so on. And with that goal in mind, we have the SPHN Semantic Interoperability Framework. And the goal is to make sense of data and add value. And the primary component of this is, of course, data semantics, where we make use of control vocabulary for concepts and value sets. And we use data standards like ATC, SNOMED, CT, ICD-10, just to name a few. And we also rely on RDF as the language for knowledge representation and use W3C standards like RDFS, SCOS, and OWL to represent the SPHN semantics. And then we also rely on other W3C standards like Shackle and Sparkle as the baseline for our quality control framework. And what this allows us to do is to enable the generation and validation of fair research data in a way that, and also have that data be available for researchers such that they can work with this data consistently across projects, systems, and over time. And so one key component is, of course, the concepts of this semantic interoperability framework. And so we have to talk about concept design. And we follow certain guiding principles that helps us in our concept definitions. And one key guiding principle is to differentiate between a process and an entity. And this is important because when you talk about observational data, it's not easy to draw a line between what is, or rather draw a line on what that data is and how to represent it as a concept. So thinking in this way helps, at least structuring how you should model your concepts. And then using that, we define the concepts along with its composed of, which is basically properties. So on the right, we see a process where we have an entity as an input and an entity as an output. But then when you go one level down to more specific representations, you have measurement with a patient as the input and a result as an output. And you again go one level down with a heart rate measurement, where you have a patient as the input to this measurement and the heart rate being the output or the outcome. And this way helps us design a lot of the concepts and also arrange most of our concepts in a coherent manner. And we use semantic inheritance and also make use of well-established standards so that we can map our concepts to other external standards like SNOMED, CT, or LOINC. And we also use value set binding to make sure that the values for certain properties are within well-recognized standards. And also we define value sets within the schema with the goal of reducing the use of strings for any property. Now, there are about 165 concepts in the SPHN RDF schema as of February this year. And we have concepts for patients that represents information about patients, demographics, medication, procedures, and also information about diagnosis, lab tests, measurements, sample and biobanking, also special concepts for oncology, intensive care, and even microbiology, and also concepts for data provenance. So just to give you an idea, if you look on the right, there's a heart rate measurement concept again, and you see that it links to a heart rate concept, but then it also links to a performer who performed the measurement, the physiological state of the patient when the measurement was taken, the medical device that was used, the body site where the measurement was made, and also the subject pseudo-identifier, which is another way of saying patient. And this is, again, the same concept, but represented in a machine-readable form using OWL and RDFS. So you see the heart rate measurement. I'm sorry if the text is too small, but bear with me. The heart rate measurement is an OWL class with a label. It's a subclass of measurement and is equivalent to SNOMED, a concept from SNOMED CT, which represents the pulse-taking procedure. And also there's a definition. And we also make use of restrictions to define how certain properties ought to be used for instances of this concept. Now, when we design the concept, the goal is extensibility, where projects can reuse the concepts and the properties we define, but they can also define their own. And they can also include additional standards that may be relevant for their subdomains. And we provide a service called SPHN Schema Forge. And the goal of the service is projects can upload their concept definitions as an Excel file. And then they can generate their semantic artifacts like the schema, the project-specific RDF schema, shackle shapes, Sparkle queries, and also HTML documentation. And concepts of general interest are then reviewed, and then especially concepts that are useful across projects are then reviewed and included in future releases of the SPHN RDF schema. Now, when I talk about projects, there are quite a few projects running in parallel, but the 4 main projects that I wanted to highlight today was the NDSs, or the National Data Streams. And these are projects that focus on infections in intensive care units, oncology, pediatric health, and also health research. And each of these projects have additional data types in addition to clinical routine. For example, genomics, metataxonomic, proteomic, metabolomics, and so on. And the goal of these NDSs is to create a single knowledge graph for reuse, where you have the routine data from the hospitals, but also you have omics data or metadata about omics, and then additional clinical data and data from cohorts and registries, and also reference data from either knowledge bases or terminologies. Now, you might have guessed by now that we've been talking about clinical routine data, but omics and all these new data types are new. And so the challenge we face is a way to represent metadata about these different additional data types. And so we first approached this by looking at just genomics, because NGS is becoming a key part of clinical routine. And we focus on designing concepts with the idea of representing information about samples, processing, assay, analysis, and then with the goal of extending the current SPHN RDF schema, and also making the concepts such that they could be extensible by the NDSs for their specific omics types. And so with that goal in mind, we collaborated with the Hive, and they will be talking about how they engaged with the various stakeholders, whether that is the university, hospitals, and the projects, to define these concepts.
Yeah, thanks, Deepak. All right, so I will tell you a little bit about our modeling approach and results to design this omics extension. So last summer, we gathered with all experts and stakeholders at a workshop to scope out the possibilities, the possible concepts that are important in omics research. So we had post-it sessions where, for instance, a clinician would say, the gene panel is an important concept for me. A bioinformatician would tell, I'm more interested in the quality metrics of a certain experiment. Another thing we did there was try to come up with a generalized omics process. and then see where every concept fits in this workflow. So with this groundwork, we continued to conduct more focused interviews with experts from hospitals, clinicians, and try to structure the domain knowledge, we call it, in a more formalized way. And from this, as you might know, the standard modeling cycle, in the end, we started coding and formalizing our model. And in the end, after this whole modeling work, we organized a final workshop where we again invited everyone to bring their own data, and then really try to apply our omics extension. And in the end, this omics extension is now released and available for use for all the partners of the SPHN. So one important point is, of course, not to reinvent the wheel, but see if there are already models that you can reuse. Of course, for data sharing, you all might know the nucleotide archives. However, these models were a little bit too generic and ambiguous for our use case. On the other side of the spectrum, there are the ontologies, and we heavily reused especially terms, for instance, from OBI to indicate experimental types, and EDAM ontology, for instance, for bioinformatics operations. And other special mention here is FAIR genomes, because they have the same intent to share clinical omics data. And we took inspiration from this model, but in the end we found out that we need more detailed expression of our experimental results. So in this slide, you see a generic overview of the concept extension. Keep in mind that the actual concept set of the SPHN is much bigger. So I will dive into some details in the next few slides. But the main important point here is that assay is central. So you could see for all omics fields that are specialized assays, for instance, for genomics sequencing assay. On the left, there are sample processing classes. And on the right, there are concepts for expressing data manipulation operations. And of course, this model is not independent. So we reused existing concepts of the SPHN. For instance, sample, data file, and gene are already coming from the existing concept set. So here's an example omics flow, as most bioinformaticians and researchers might recognize it. So from left to right, you start always with a sample that's analyzed, outcomes from data in the middle. Data is subsequently processed, and in the end you end up with a result. So here I took some instantiation. This is an example with the property of attribute values in red of the first part. So what you see here is a sequencing assay, whole genome sequencing using some Illumina instrument that analyzed some blood sample, and out comes some FASTQ file. Of course, this FASTQ file serves it as input for the next step. In this case, there's an example of variant calling using Dragon pipeline, some reference genome in this case, and the final result. So you can really imagine that a genome screening center uses this way to represent and share data with the wider consortium. And this brings me to the conclusion of this work. So we created the omics extension for the SPHN. That's suitable for sharing genome of omics work with a focus on genomics, but they're extension point for other areas. And we aligned heavily with existing ontologies. So this improves interoperability, for instance. And we really built this as a collaborative approach. So of course, we presented it together, but there's a whole consortium that contributed to this work. And with this, I would like to pass the microphone.
Thank you, LK. Thanks. So yeah, that means we designed the concepts, and these concepts are already part of the 2024 release. So the goal now is to see how the NDSs are using this concept to define additional subsequent concepts, and also how data providers are providing the data given the specification that we have made. And then also to expand the focus to other omics types, because for now it's just genomics, but then we want to expand for proteomics, metabolomics as the request comes from the projects. And then concepts that have general importance or concepts that are valid across projects can be incorporated in future releases. Now, maybe, in a setting like this, I think this slide is important because SPHN as an initiative is funded till 2024. So we are winding down, but then we will be coming back, or we will be continuing our work in 2025 as a National Data Coordination Center. So the work that we're doing will continue for the coming years. So it is not an existential risk. I just wanted to highlight that because in previous talks, we have said that the funding is still 2024. So I thought we just want to set the record straight. And with that, I'd like to thank the members of the Personalized Health Informatics Group at the SIB, our collaborators at the Hive, and also the SPHN National Steering Board, Advisory Board, task forces, working groups, and also the hospital workforces who have worked tirelessly with us with the concept design and also a lot of the technical aspects, and also the management office. And then we can open the floor for questions.
Thank you very much. So we have one question on Slido. I don't see right now hands raised. So let's go to start with the one from Slido.
Hi.
Hello.
Does your data model look into the content of BCF? Example, the location of a variation?
So we do have concepts for genetic variation, but then we are expanding it further to accommodate more information and different variant types. So it's a work that is ongoing. And also we are looking into the idea of variant interpretation, variant descriptors, and how that information is represented in the phenopackets specification, because that has been very useful from a clinical perspective to tie in the data points to a patient and their medical history. And so that is kind of the focus we want to have, because there are plenty of data models out there that just talks about representing variants in genomics in general in research. But when you want to tie it to clinical, it's a bit more challenging. So yeah, the discussions are ongoing.
Hi, I have a question regarding the sequencing pipeline. So in the vocabulary for it. So if you look really carefully into sequencing data, then you see it's not like clear in many cases. So you have a lot of ambiguities. So how do you encounter this problem of base pulling in your vocabulary? Or do you have like a quality measurement for the quality of the sequencing in your terminology?
Yes, good question. We mainly express data at a metadata level. So actual configuration output that's stored and linked in files.
Yeah.
For quality, yes, we provide concepts for expressing quality metrics, either linked to sequencing experiment, but could also be linked to processing steps, sample preparation steps. These also have some quality criteria output. We provide a quality metric concept for that, where you can choose from certain values, for instance, threat score, or to express these things. Yeah.
Okay. Thank you.
stand in the way of the camera. So thank you for a wonderful wonderful talk. And I have a question that sort of continues on what you mentioned about because you mentioned pheno packets, and you mentioned that you looked at ENA and EGA. So I guess for when you're, and we have that in the rare disease community as well, that when you go into the realm of genomics, there's sort of, you can't go around the Global Alliance for Genomics and Health. So I was curious if you, so we're also looking into that. So how do we become interoperable with the standards that they are working on? And I was wondering if you have done such efforts as well to be, how to be interoperable with those standards.
So we have looked at briefly at the GA4GH, of course, because they've been doing a lot in terms of the models. But then a lot of what comes as the outputs are also for more research-oriented data structures and data sources. But the outlier, or maybe the exception being phenopackets, right? And we want to engage with them more because there's a new working group for categorical variants. There's another working group for variant representation. And so a lot of these discussions are happening in parallel, and maybe, yeah, it might be good to see where that goes, and also to learn from your experience, because you have been doing this for several years, where you want to represent variants within the context of the rare disease community and how that's worked so far. Maybe phenopackets work for you out of the box, or maybe there was things that you had to do around it to tie it together to other things.
Yeah, and actually we worked on semantic phenopackets to make an ontological version of that, and we want to update that.
Okay, then we should.
And yeah, definitely we can collaborate on that. Actually,
Wonderful.
maybe even here we have some things set up here. I'll talk
Great,
to you about.
thank you.
Thank you.
Looking forward to that. One more question?
Thank you for a great talk. I had a question about the underlying tools that you may use in the sequence analysis. So for instance, variant calling. Most of this software is probably not FAIR. And my question was, do you expose this sort of underlying quality or underlying parameters of these programs in your data model? And how do you do it? What is your approach?
That is a wonderful question. So that comes to the part where we want to expand the concepts to workflows, right? Because when you run some analysis, it has to be a multi-step workflow, and then that has to be represented. So we haven't gotten there yet. That's because the data providers who are interested in providing the data have not yet agreed upon how to provide that information. But yes, preliminary discussions suggest that we should keep track of the parameters, the tools, their dependencies, the versions. And if there is a protocol for running that analysis, that should also be available. So yeah, I think it's very much an ongoing discussion, and very sometimes heated discussion. So I completely understand.
I have a question from one of the virtual
Sure.
attendees. When you integrate other omics types of data, are you planning to integrate it into the same knowledge graph or build interoperable domain-specific knowledge graphs?
Yeah, I think this might come to the NDSs, right? So the National Data Streams, on how they prepare their knowledge graphs, because for some types would be more important, some omics type would be more important than the others. But the schema that we have, it could be the core SPHN schema, or it could also be project specific. So it becomes a very project and thus domain specific option. I think we have to really evaluate, right? So because our inclusion criteria, which is quite flexible to be honest, but still we try to see if there are concepts of importance across projects, then we bring it into the core. And that becomes much more easier as a specification for data providers to give that information. And our data providers are the university hospitals. So we have to make sure what we put in the main specification is consistent, and there's no ambiguity, or they have problems at the data when they provide this data.
Yeah, I'm not taking the word from anyone. I just wanted to make a comment. It's kind of a follow-up from the previous question about the software quality. And you mentioned workflows. So the suggestion would be for you to look at the FAIR for research software, FAIR principles, and also for the work around FAIR workflows. None of the metadata so far as I know, really include quality measurements of software. There is the initiative of the Matching Actionable Software Management Plans that it is already working on top of schema.org. And there is an early work from the EOS task force on software infrastructures. There was a subgroup working on quality metrics for software. Out of a list of kind of 100 or so quality metrics, only a bunch of them were mapped to the FAIR principles and to the machine actionable software management plans. But maybe this is useful to look at for the case of software and software quality.
That would be good, because that might help also the discussion that we're already having. And we also got a lot of suggestions from the reviewers who reviewed our abstract. They suggested to look at Nextflow and common workflow language. So there's already a lot of work there. So yeah.